Hi class, in this recording we're going to continue our discussion of biomolecules within the human body, particularly the macromolecules. So let's dive in and talk about lipids. When we think of a lipid, lipids are a category of macromolecule in our body that are hydrophobic, and it's the only hydrophobic. So if it does not like mixing with water, I want you to think of it as a lipid. And as we think of these lipids, they also have a very high hydrogen rate to oxygen ratio. So they have a lot more hydrogen in them than they do oxygen. And this is one of the reasons why they are hydrophobic. They typically tend to have large nonpolar regions on them. Something that's notable is that lipids have more calories per grams than carbohydrates, proteins, or nucleic acids. So they tend to be more energy dense. And as we look at our lipids, they are going to be broken up into three broad categories: the triglycerides, the phospholipids, and the steroids. So let's talk about triglycerides. When we think of triglycerides, I want you to think of lard or just fat. A triglyceride, as the name implies, is going to have three of something. What does it have three of? It has three fatty acids. And these three fatty acids are going to be attached to one glycerol head. And as we think of triglycerides, this is how we typically store most of the excess energy in our bodies for long-term storage. Now, as we look at these triglycerides, they can be made through dehydration synthesis, and they can be broken down through hydrolysis. So as we think of this process of breaking down fats in our bodies, we need to use up water in order to break down the chemical bonds that hold fat molecules, triglycerides, together. At room temperature, if the triglyceride is liquid, we call it an oil. If it's solid, we call it a fat. Typically, liquid triglycerides are going to have polyunsaturated fatty acids and are usually going to be from plants. When we think of solid triglycerides, they are typically going to have saturated fatty acid tails, and those are usually going to be from animals or sourced from animals. As we think of these triglycerides, they are for long-term energy storage, but they also help us with insulation and shock absorption. The tissue type that we use to store these triglycerides is going to be adipose tissue. Adipose tissue is fairly complex, and we'll talk about it more when we get to histology, but I just want to emphasize now, we store triglycerides in adipose tissue. So as we look at trans fats versus cis fats, these are going to be all based on those fatty acid tails. All the fats here, these trans and cis fats, are unsaturated fats. What we mean by that is that we have a double bond present between two carbon atoms. And this is a skeletal diagram down here. If you haven't taken organic chemistry, you probably aren't very familiar with the skeletal notation. We're not going to spend a lot of time talking about it because this is A and B. So as we look at trans fats are going to have the carbons on opposite sides of the carbon-carbon double bond. And they will typically have a more linear orientation to their structure. One of the things about trans fats that's notable is that trans fats are chemically at a lower energy state, so they're more stable and harder to remove. Something else that is notable is that trans fats are not produced in nature. They're only produced artificially when we catalytically hydrogenate unsaturated fats. So when we think of trans fats, and if you have partially hydrogenated oils, those partially hydrogenated oils by default will have trans fats present in them. Because they're at a chemically lower state or lower energy state, they're harder to remove and more likely going to be correlated, not causative, but correlated with individuals that have heart or cardiovascular diseases. When we think of cis fats are going to have the carbons on the same side of that double bond, and because the carbons are present on the same side of the double bond, those cis fats are going to have a bent configuration. That bent configuration means that the cis fatty acids are not going to be able to be stored as tightly and compacted as tightly, and consequently are going to be associated with lower energy densities and typically being more of a liquid or oil at room temperature as opposed to a solid or fat at room temperature. Something else that's worth emphasizing with cis fats is cis fats are made in nature and they have a higher energy state than trans fats. Because they have that higher energy state, they have more stored chemical energy in them, they are easier to break down. As we look at phospholipids, a phospholipid is a modified triglyceride. So we still have that glycerol head. We can still have two fatty acid tails, but then instead of the third fatty acid tail, we're going to have a phosphate and nitrogen containing head group. And this head group here is usually just diagrammed out as a circle. And as we're looking at phospholipids, they're typically going to be used to make up membranes, like the cell membrane within our body. And we refer to these molecules as amphipathic. And this is a pretty cool property. The fatty acid tails are nonpolar, so they are hydrophobic, but the phosphate head up here is polar, and because it's polar, the phosphate head is going to be considered hydrophilic in nature. So phospholipids serve as an intermediate or a surfactant in that they can be dissolved in water and they can be dissolved in oil. So these are the molecules that serve as an interface between, that may serve as an interface between a chemical compound that is nonpolar but needs to be dissolved within water or an aqueous solution. Another way to think of these is you can think of them as natural soap, because they can take something that doesn't like to be dissolved in water and bind to it and then help to dissolve it within water. As we look at steroids, steroids are a category of lipids that have 17 carbon atoms within 4 separate rings. So if you ever see this 4-ring structure, I want you to think of steroids. All steroids are going to start with the base cholesterol. So that's our parent molecule. Some cholesterol in our body is from dietary sources. The vast majority of the cholesterol within our body, it comes from our own bodies manufacturing it. And as we look at cholesterol, not only is it going to be important because it's a precursor for the steroids in our bodies, it's also important because it's used to modify the fluidity or viscosity of the cell membrane. If we have increased concentrations of cholesterol within our cell membranes tend to be more viscous. And if we reduce the concentration of cholesterol in our membranes become less viscous. Something interesting that happens is even within your own body, as you acclimate, as you become cold weather acclimated, you can temporarily or slightly reduce the concentration of cholesterol in your cell membranes. And as you become warm weather acclimated, you slightly increase concentrations of cholesterol within the cell membranes of your bodies. That cholesterol can be used to make a variety of steroid-based hormones, such as cortisol, progesterone, estrogen, testosterone, and a variety of bile acid salts. That's all we have for this recording. If you have any questions, please feel free to post them on the class discussion board or to shoot me an email. And as always, happy studies.